Good evening, everyone. Uh, Welcome you all to the live interaction session of NPTEL. Today, we'll be discussing some sample questions on recombination, that is the week 3 lecture series. So, before that, let's wait for about 5 to 6 minutes so that people can join and then we will start. Okay. We will start from 6:10. Just wait for 5 minutes. I guess I am audible to all of you. Am I audible? Okay, okay. Thank you. Thank you for confirming. Okay, good evening everyone. Welcome you all to the NPTEL TA session of week 3. So let's start our sample question series. If you have any problem regarding the question or screen or my voice, then please let me know in the chat box. Otherwise, I won't be able to understand if I am audible or if my screen is visible or not. Okay. So this is the first question. What is termed as a guardian of genome integrity? First one is cell division, mutation, DNA damage, or homologous recombination. Please try to answer in 2 minutes. Then we will discuss. Please answer. So, as you have answered, the guardian of genome integrity is homologous recombination. Why? Let's see. So, guardian of genome integrity means who, which mechanism maintains the genome of an organism. So, first is cell division. Cell division, what it does, it generally divides the cell, and during the cell division phase, replication of DNA occurs, and during that replication process, crossing over and other things happen, which changes the genome of an organism. It might be haploid or diploid of the genome size, but during replication or cell division process, the genome or gene sequence changes, or you can say if in a zygote case, but in case of this somatic cell division, it doesn't change, but we can't say completely that it maintains the genome integrity. Okay, next is mutation. So what is mutation? Mutation occurs due to any endogenous or exogenous factor, and this mutation changes the DNA sequence. Okay, so if the DNA sequence is changed, that means genome is also changing, so it is not the answer. Next is DNA damage. As we have already discussed DNA damage in our previous section, so DNA damage generally happens due to lot of factors, and it changes the nucleotide sequence or genome, or destroys the genome integrity. So answer is 4, that is homologous recombination. So what is homologous recombination? It is termed as the guardian of genome integrity as it acts to repair DNA damage. So if there is one DNA damage occurs in the genome, homologous recombination is a important mechanism which repairs this DNA damage. If you recall it, we have discussed about recombination repair in our previous session, where we studied about how this recombination repair mechanism repair the DNA damage. So this is an example of homologous recombination also. And this homologous recombination also promotes the pairing between identical or nearly identical DNA sequences. Please understand this point, and it is important. because generally it asks in questions like in four options, MCQ questions. So what is the answer if it is between identical or non-identical sequencing cross repair. So please remember it that the pairing occurs between identical or nearly identical DNA sequences, nearly identical or identical, and the subsequent exchange of genetic material between them. Okay, we will be studying about the whole recombination process in this session. So please wait for that. Okay, I think everyone is clear with homologous recombination now. We will move to our next question. Next question is, DNA ends resulting from a double strand break is processed by a multifunctional enzyme complex called REC-BCD, DNA polymerase, RNA polymerase, or gyrase. Please answer in 2 minutes, then we will discuss. If you have any doubt regarding any question or anything, then please let me know. I will clear your doubt. Everyone, please try to answer this question. Okay, so you have answered different type diversity like there is okay. Some of you have answered gyrase, DNA polymerase, or REC-BCD. Anyone want to answer? Other people, please try. If you want to answer, then I'll wait. Okay, let's discuss then. So, first we need to understand the function of each and every enzyme presented here so that we can find out the correct answer. Okay, so let's see. If you don't know the function of any enzyme complex here, then first try to uh, just understand the enzyme complex which you know. Okay, everyone knows about this DNA polymerase, right? DNA polymerase, what is its function? What is the function of DNA polymerase? So DNA polymerase is the enzyme required in the DNA replication process, and it adds nucleotides, right? It adds nucleotides during this replication process. Okay, DNA polymerase is an enzyme required for DNA replication process, and it adds nucleotides and increases the chain length. So it can't be able to repair a double strand break. As you might have mm, misunderstood, like DNA polymerase has different subunits in case of eukaryotes, and it just does this proofreading mechanism. Okay, not DNA double strand break repair. It helps in proofreading. Like if one base is substituted by one incorrect base, then it just removes that incorrect base and ligase then joins the DNA. Understood? So DNA polymerase has proofreading mechanism, but it can repair a double strand break. Next one is RNA polymerase. So what is the function of RNA polymerase? RNA polymerase is required for the transcription process. The same thing, it adds nucleotides and increases the chain length. Okay. Next, gyrase. Gyrase is an enzyme which is present in prokaryotic organisms like bacteria, and it is a family of topoisomerases. So as you have discussed in the previous session, gyrase or topoisomerases, they just helps the superhelical coil of DNA to remove. Okay, they just opens up the DNA. They don't have any role in repair mechanism. Okay, so as we have known the function of these three enzymes, so now we can easily select the correct answer, that is REC-BCD. Okay, so DNA ends resulting from double strand break is processed by a multifunctional enzyme complex is called REC-BCD. So what is REC-BCD? Let's see. REC-BCD is a sequence-regulated bipolar helicase nuclease that splits the duplex into component strands and digests them until it encounters a chi site, which is a recombinational hotspot. So here it is important, helicase nucleus. It has two functions. It can be act as a helicase and then nuclease. It has two functions. Okay. Please try to understand. If you have any questions, then please let me know. And this nuclease activity, so everyone knows about the nuclease activity. It just cuts the DNA. Okay, it cuts the phosphodiester bonds and moves out or the nucleotides. And nuclease activity is then attenuated, and REC basically loads REC-A onto the 3' tail of DNA. So let's see. This is the structure of REC-BCD. Just a second. This is the structure of REC-BCD. So as it is colored differently, anyone wants to say anything? Or okay. Okay, let's see. Here it is the structure of REC-BCD. So it is colored differently for different subunit. So this is the REC-B site, this is REC-C site, and this is REC-D site. This is a complex protein with three subunits. Okay, and it is the DNA, how they hold the DNA. And this is the 3' single strand, SS strand, buried, and F' 5' SS strand. And this is called nucleus domain and tunnel. It is visualized clearly in this figure. Next, move to the function. What it does actually. First, REC-C recognizes the chi site. So chi site is an hotspot of recombination in case of prokaryotics like bacteria. Okay, so the REC-BCD is the pathway of homologous recombination in case of prokaryotes like bacteria. Okay, we are studying about the homologous recombination process in case of prokaryotes. Don't be confused with the eukaryotic protein and prokaryotic protein. Let's see. This REC-C recognizes the chi site, which is the recombination hotspot Okay, and this hotspot has certain residues or certain nucleotides common or conserved in case of prokaryotes. This is the sequence, 5 days to 3 days site. After that, rec C signals rec D to stop. Rec D is an helicase. Okay, it's a helicase. So it just opens up the DNA. Next, when stopped, rec D signals rec B to cut DNA. Rec B then cuts the DNA and rec B cuts where it is and continues unwinding of DNA leading rec A. So it is the function of rec B C D complex. There is one protein rec A which just adds the nucleotides. Okay. So if you have any doubt, then please ask me. Next, move to the function, how it repairs the DNA. This is the REC-BCD pathway of homologous recombination in prokaryotes. So, as you have already seen the REC-BCD complex, if there is one double strand break occurs inside any DNA, REC-BCD detects it and attaches at that point. Then it cuts the DNA, as you can see here, right? It cuts the DNA, right? Cutting, cutting, cutting. After that, here ATP is required. Remember this, ATP is required for each and every process. After that, when the REC-BCD complex reaches the chi site, that is the sequence written here, it reaches the chi site, then you can see that this 5-day strands remains intact. 5-day strands remain intact, only the 3 days to 5 day strands cutting happens. See here. Cutting of 3 days to 5 day strand happens and this 5 day strand remain intact. After that, after cutting that 3 days to 5 days strand, ray K filament is added. The ray K filament, it contains the proteins which adds nucleotides and act in a way where it increases the chain length. So it has been cut. Okay. Now using this 5 days to 3 days site as a template, it repairs the DNA or adds nucleotide sequence and increases the chain length. Like this, the whole process happens in case of REC-BCD pathway. I guess everyone is clear with REC-BCD homologous recombination. If you say, I can repeat. Anyone have any doubt? Do you want to repeat this? Or everyone is clear? Is it clear to everyone? Okay. Am I audible? Okay, okay, fine, fine. Thank you. Uh, if you have any question, then ask me. Otherwise, I will move to next question. Okay. So I think everyone is clear with this REC-BCD pathway. Next, move to our next question. That is, a chi site is option 1, replication hotspot, DNA damage hotspot, recombination hotspot, and transcription origin. I think I just discussed about it. Please answer this question, then we will discuss. So, as you have answered, Chi-X site is a recombination hotspot. Okay, it is a short stretch of DNA in the genome of a bacterium near which homologous recombination is more likely to occur than on an average across the genome. Okay, for this reason, Chi sites are also referred to as recombination hotspots. So this is the reason why it is called as recombination hotspot, because it has the more chances of recombination at this point other than the different genome sequence. Chi sites serve as stimulators of DNA double-strand break repair in bacteria, and its sequence is unique to each group of closely related organisms. In bacteria E. coli and Salmonella, the core sequence is this: 5 days GCT, GGT, GG. So this sequence is conserved for E. coli and Salmonella. In addition, it includes about 4 to 7 nucleotides to the 3-day side of core sequence, which are important. So I guess it is the whole thing about Chi-X site, which is responsible for recombination or homologous recombination in case of bacteria. Everyone is clear? So let's move to our next question. In the classical non-homologous end joining pathway, which protein acts as the initial DNA end binding factor? Ku heterodimer, REC-BCD, XRCC4, or DNA PKCS? Please try to answer in 2 minutes. Then we will discuss the NHEJ pathway along with different proteins. Please everyone try to answer. Okay, so let's discuss. So NH stands for classical non-homologous end joining pathway. Okay, this is classical non-homologous end joining pathway in which the protein acts as the initial DNA end binding factor. Answer is Ku heterodimer. Okay, let's see. Because you know that REC-BCD is responsible for DNA binding in case of prokaryotes, and here we are talking prokaryotes for homologous recombination, and here we are talking about NHEJ pathway. Okay, so let's see this canonical non-homologous end joining CNHEJ pathway depends on Ku heterodimer and DNA PK, that is the catalytic subunit, which together form the DNA PK holoenzyme. Ku is a fast responder at double-stranded breaks and provides a docking site for DNA PKCS. So this schematic representation, here you can see here that just a second. So here one double strand break already happened. So to detect that, first comes the Ku7080 heterodimer. Okay, it's an heterodimer. Then it binds to the DNA site and creates a docking groove for DNA PKCS. This DNA PKCS stands for the DNA PK catalytic subunit. Okay, and after binding the DNA PKCS, mRNA, CTLP, Artemis, etc comes here, and binding of these molecules and protein complexes happens, which leads to repair of the DNA double strand break, and at the end lipase 4, XRCC4 and XLF like proteins, they just rejoin the structure. Okay, this is just a simple schematic representation of NHEJ pathway, not complete strands, how it is mRNA, what its function, CTLP, Artemis, like that. Okay, it's already written here. You can read that. Unlike mRNA, which can bind internally, Ku requires free DNA end. So here after the break of double stranded break happens, it needs to be some 5 days or 3 days overhang where Ku heterodimers binds and cannot associate with most blocked ends. Okay, and several nucleus including tyrosyl, DNA phosphodiesterase 1 2 and Artemis can remove hairpins, damaged bases or protein DNA adducts. So this Artemis and TDP 1 or 2, they can remove the hairpin structure formation, damaged bases or protein DNA adducts. Means if some during replication or some kind of transcription or like that, if protein DNA complex formation happens, that also creates a problem for continuing the process of replication or transcription. That's why some enzymes like TDP12 or Artemis, they help in resolving that problem. Okay, then DNA ends are processed by additional enzymes and they are joined by LEAK4, XRCC4 and XLF complex. Okay, these are a complex of molecules which help to rejoin the DNA ends. Okay, I guess everyone is clear with NHEJ pathway where Ku heterodimer plays an important role by binding the open DNA strands. Okay, and its main function is creating a docking site for DNA PKCS where DNA PKCS can bind. Okay, I guess everyone understood. Let's move to our next question. Question is, site-specific recombination between sites located on the same chromosome in a head-to-tail orientation may result into excision, inversion, integration or recombination. So now we talked about homologous recombination of REC-BCD pathway, then non-homologous end joining, and now the question arises from site-specific recombination. We'll be talking about it shortly. So please try to answer this question. Then we'll talk about it. Okay. Everyone, please try to answer this question. So, uh, you have answered excision and some of you have answered recombination. So let's see. First, try to understand the question. Okay. Site specific recombination between sites located on the same chromosome. Okay. Same chromosome it is asked in a head to tail orientation result into. Okay. Let's see. Let's first understand the terms. So, site-specific recombination encompass a group of processes in which DNA molecules are rearranged by breaking and rejoining the strands at specific points. So, this is the definition of site-specific recombination. Okay. So, for the first case, if you see here, this is the gene 1, gene 2. If they are added, it's a head or tail orientation. Okay. This is the candidate gene and it is the head or tail orientation of a gene. So if head to tail orientation recombination happens, so it will remove the candidate gene like this to this structure only. Can you see this part? It is uh, in orange color. So it will be removed and it's called excision. So only this part will remain and this part. So it will be called excision. So this is called head to tail orientation of site specific recombination. Okay. And after this head to tail orientation of site specific recombination, some part of the gene is removed and that's why it's called excision. Okay. Next, insertion or integration. If there are two genes, candidate genes, there are two genes, head to tail orientation. If insertion happens, now you can see here, 2 is replaced by 4. So this is called insertion or integration. Okay. Next is inversion. Inversion means very simple. It just inverts the DNA or gene. Okay. Like it is head or it is tail. Now it just inverts. Now it is head or now it is tail. Okay. So I guess everyone is cleared with the terms excision, insertion, and inversion. And other thing, some of you have answered recombination. So recombination happens when there are genes or exchange of same or nearly same DNA sequences occur in two two alleles or two chromosomes that needs to be there. Understood? It is asked on same chromosome, so it can't be recombination. I guess everyone is clear with excision, inversion, integration, and then recombination. Don't get confused. If it is our same chromosome, then go for these options first. Okay. Okay. The next question is the tyrosine type recombinase catalyze 180-degree rotation and rejoining of cleaved substrate DNAs, formation of a holiday junction intermediate, single strand break repair, or translation. Please try to answer this question in 2 minutes, then we will discuss. Everyone, please try to answer. Okay, let's discuss then. Okay, so before going through the options, we need to understand what is tyrosine type recombinase and how it is doing its work, or what is its function. Okay, let's see. I have added some text here because you need to go through the theory first to understand the function of tyrosine type recombinases. So enzymes which catalyze site-specific recombination between two specific DNA sequences and mediate DNA integration, excision, resolution or inversion are called site-specific recombinase. So what are the site-specific recombinase? And these are the terms which need to be understood by each and everyone, like integration, excision, resolution, or inversion. I guess we have discussed the terms. Everyone is clear with it. Next is site-specific recombinases, which play a pivotal role in the life cycles of many microorganisms, are classified as tyrosine type or serine type recombinases based on whether a tyrosine or serine residue mediate catalysis. Okay, so this site specific recombinases, by the name suggests, you can understand that it is a protein. So protein is made up of amino acids, and when the tyrosine residue functions or mediates the catalysis, it's called tyrosine type recombinase. And when a serine residue functions or mediates the catalytic process, then it is called serine type recombinase. Okay, and they are further subdivided into two subfamilies, each based on following criteria. Tyrosine type recombinases are subdivided by the recombination directionality. Recombination directionality and serine type recombinases are subdivided by the protein size. Okay, and then tyrosine type recombinases catalyze the formation of holiday junction intermediate and resolution. So what they do, they catalyze the formation of holiday junction intermediate and leads to resolution of the crossing over. This catalytic mechanism of serine time recombinases include 180-degree rotation and rejoining of cleaved substrate DNA. So this holiday junction generation happens when homologous recombination happens there in a eukaryotic organism, and after and this junction needs to be resolved, and for that tyrosine type recombinases are mostly preferred. Okay, I guess everyone is clear. Okay, so answer is formation of a holiday junction intermediate. So this tyrosine type recombinases, they are site specific recombinases and they catalyze the holiday junction. Okay, and 180-degree rotation, as you have seen here, it is catalyzed by serine type recombinases. Okay, then single strand break repair, I guess we have talked about it, base excision repair, nucleotide excision repair and all that, but no recombinase is responsible for single strand break repair. And the translation process, there are ribosomes and different enzymes responsible for translation, not recombinases. If you get confused with the options, so first try to eliminate the options first which you understand or which you know the function about that particular enzyme or thing. Okay, first try to eliminate them, then you will get the answer easily. Okay, I guess everyone is clear. Let's move to our next question. The integration of bacteriophage lambda into the Escherichia coli chromosome is an example of homologous recombination, non-homologous recombination, site-specific recombination, and DNA repair. I guess we are familiar with all the terms. We have discussed all the terms so far. So please try to answer it. Then we will discuss. Everyone, please try to answer. Okay, so everyone has answered three. That is site specific recombination. Great. This is the correct answer, right? So, what is the definition of homologous recombination? What is the meaning of homologous recombination? We just discussed at the starting of this class. Can anyone please tell what is homologous recombination? So it's a double strand break repair mechanism, okay, and okay, that maintains the genome, okay, DNA damage repair system. DNA, it repairs the DNA damage, so it's called the guardian of genome integrity. Then we discussed about non-homologous recombination, which we discussed in case of eukaryotes, and then comes the site-specific recombination and DNA repair. So integration of bacteriophage lambda, integration term is very specific to site-specific recombination, as we have already discussed, right? So when the integration of bacteriophage lambda to Escherichia coli or chromosome happens, there need to be particular DNA sequence which gets recombined or gets exchanged, right? Let's see the mechanism. So this is a circular chromosome of lambda bacteriophage, and this is our gene of interest. So we know the sequence of the gene, right? For site specific recombination, as the name suggests, it is specific to particular site with known DNA sequence, right? For homologous recombination and non-homologous end joining, it is one of the DNA repair mechanisms, and it generally happens when there is one double strand break occurs in the DNA. Okay, so bacterial chromosome is here, and this is the attachment site sequences. Integrase binds lambda integrase protein complex, it is binds here, so it includes the specific DNA sequence of lambda bacteriophage and bacterial chromosome. Okay, next, catalysis of double strand break happens, as you see here, breakage and rejoining. This break is there, and then this part is rejoined. Okay, look at the figure. And then integrate dissociates happen, and bacteriophage DNA integrated into bacterial chromosome. This is a simple schematic diagram of how bacteriophage DNA can be integrated into bacterial chromosome. The other enzymes which are responsible for this mechanism has not been shown here, so you can go through the mechanism in your slides only. So it is just a simple representation of how integration of this bacteriophage lambda DNA occurs with bacterial chromosome. Okay, I guess everyone is clear with site specific recombination between this bacteria and bacteriophage. Okay, so let's move to our next question. 180-degree rotation and rejoining of cleaved substrate DNAs is catalyzed by serine type recombinases, tyrosine type recombinase, both serine type and tyrosine type, or FLP recombinase. Please try to answer this question in 2 minutes. Then we will discuss. So everyone, most of you have answered one, that is serine type recombinase, and it is the correct answer. Great. I think everyone is clear with site-specific recombination then. So we just discussed that site-specific recombination is carried out by two recombinases, two types of recombinases, serine type recombinases and tyrosine type recombinases, right? So this tyrosine type recombinases, they help in catalysis of holiday junction. Okay, and this serine type recombinase, they helps in 180-degree rotation and rejoining process. Let's see how it works. Okay, so this is the schematic diagram of serine type recombinase catalyzed recombination. So these are the two DNA strands or two chromosomes present here. Okay, two chromosomes are there, and after cleavage happens, because it is already discussed that it is a type of site specific recombination. So there are particular sites where we need to where we want to exchange the chromosome between them. Okay, so exchange of first need to be cleavage, then exchange of DNA strands happen, as you can see here. Okay, this blue one comes here from to this. This is the exchange process. After that, ligation happen. Ligation means just joining of the closed open strands. Okay, ligation happens, and then 180-degree rotation happens. So, and P stands for phosphorylserine linkage. So each hydroxyl group of catalytic serine residue in all four recombinant molecules act as nucleophile, attacks the substrate DNA and forms 5 days phosphorylserine linkage. So here this bond is shown as red color. It is called phosphorylserine linkage, and it is formed by the hydroxyl group OH group of serine. Okay, and it acts as a nucleophile. I guess everyone is clear with nucleophile and electrophile. It acts as a nucleophile and then attacks the substrate DNA and forms a phosphorylserine linkage. Understood? Then two of four recombinases molecules that are covalently linked to different substrate DNAs rotate with respect to the other pair of recombinase and relegate their attached 5 days phosphate to 3 days OH of the unrotated DNA. Simple term, as it is shown that first exchange of DNA strands happen between two chromosomes. After that ligation happen, and this rotation happen in a 180-degree orientation. Okay, 180-degree rotation happens. Okay, so serine type recombinase catalyzed recombination includes 180-degree rotation and rejoining of strands. Understood? I think everyone is clear. So let's move to our next question. This is a very simple question, but still it is asked in a NET-LS or GATE exam for easy section. Who proposed holiday model for homologous recombination? Govind Khurana, Louis Pasteur, Robin Holliday, or Niels Bohr? It is quite simple, I guess. So please try to answer. Everyone can answer, I guess. So please try. So yes, Robin Holiday is the correct answer. Okay. Initially, homologous recombination or homologous end joining was proposed by Robin Holliday to explain the DNA strand exchange mechanism in gene conversion events. Okay, in the 1970s, HA was first observed via electron microscopy in E. coli, providing conclusive evidence for the existence of homologous joining in organisms. So this has been shown first in a prokaryotic organism. After that, it has been discovered in case of eukaryotic organism also. Okay. So the next question is here. Name the phenomena of allele replacement during recombination and DNA repair. Is it replication, mutation, transversion, or gene conversion? Please try to answer. Then we will discuss each and every term. So many of you answered C or D. It's D, I guess. Each and everyone has answered D. So, so answer is gene conversion. Very good. So, first we need to understand each and every term, right? So, replication. What does replication mean? What is the meaning of replication? Okay. So, replication is the formation of new DNA, right? Replication is a mechanism which includes the formation of new DNA strand with the help of several enzymes. Mutation, when there is a change occurs in the DNA sequence, it is called mutation. and this mutation may be happen due to exogenous factors like UV rays, ionizing radiation, or endogenous factors like replication error, like that. Okay. Next is transversion. Okay, some of you answered transversion. So transversion is a type of point mutation. Okay, transversion. So point mutation, it is hard to write, but let me. So point mutation, it is of two types, that is transition and transversion. Okay, so point mutation is of two types, that is transition and transversion. In case of transition, the purine nucleotide bases are just mutated to purines only, or pyrimidines are mutated to pyrimidines only. In case of transversion, purines nucleotide bases, they mutated to pyrimidines, or pyrimidine nucleotide bases, they mutated to purines. So if a mutation, point mutation happens between purines only, that will be called as transition. And if purine to pyrimidine or pyrimidine to purine mutation happens, then it will be called as transversion. So transversion is nothing but a type of point mutation. And the question here is phenomena of allele replacement during recombination and DNA repair. It is called gene conversion. Let's see it, or discuss it clearly. Okay. Gene conversion is the phenomena of changing allele forms during recombination and DNA repair. So this is some schematic diagrams of gene conversion, reciprocal homologous recombination, and nested DNA inversion. Express DNA, it is as shown here. This is the schematic diagram of expressed DNA and a silent capsule. So duplicate homologous recombination leads to complete gene conversion. So here you can see, right, the gene conversion happens, and this expressed gene move to the silent capsule. Complete gene conversion happens, and this part is combined, like this green and sky blue part has been combined here. Okay, and it is called segmental gene conversion. If some segment of gene has been conversed, so then it will be called segmental gene conversion. If complete gene conversion happens, then it's called complete gene conversion. Okay. Next is reciprocal homologous recombination. It is an express gene and it is silent capsule. Reciprocal recombination leads to change of gene reciprocally, as you can see here. It is quite clear for everyone, I guess. Okay. Nested DNA inversion. Inversion means, as you have already discussed, here you can see it has been inversed like this, and a new genotype has been formed. Okay, inversion happens between this region, and a new genotype has been formed. Okay, so everyone is clear with gene conversion and nested DNA inversion. Let's move to our next question. Name the protein which is involved in homologous recombination in E. coli. Is it topoisomerase, gyrase, helicase, or REC-BCD enzyme? Please try to answer. Everyone, please try to answer this question, then we will discuss each and every term. We have discussed this question before. Please try to answer. So let's move to the discussion part. Great, everyone has answered correctly. This is REC-BCD enzyme. I am happy that everyone is clear with homologous recombination in prokaryote like REC-BCD pathway, right? So we need to discuss the term topoisomerase, chirase, helicase, and REC-BCD enzyme. Okay. So what is the function of topoisomerase? What it does? Is it required for DNA replication, transcription, or translation process? Do we require topoisomerase for translation process? Can anyone answer? Do we require topoisomerase enzyme for translation process? Great. So, this, yes, yes, very good. You have answered correctly. So this topoisomerase enzyme is responsible for relaxing the superhelical structure of DNA. So, so this topoisomerase is, as it is required for relaxing of superhelical structure of DNA, so it is required for replication and transcription processes also. Okay, not translation process, because translation process is done by the ribosomes with the help of tRNA. Okay, no role of topoisomerase is there because no DNA is involved in the translation process. Only the RNA products which are produced from the transcription process is used for translation or production of proteins. Okay, so next is gyrase. So gyrase is a enzyme in the family of topoisomerases which is responsible for the same function, that is relaxing the superhelical structure of circular DNA in case of prokaryotic organisms like bacteria. Okay, next is helicase. Can anyone tell me what is the function of a helicase? What is the function of helicase? Please try to answer. I guess everyone know it. Simple, great, great. So unwinding the DNA or separating the two strands of DNA, right? I wanted to listen it from you. That's why I waited. So helicase is the enzyme responsible for unwinding the DNA or separating the two strands of DNA. As we have already discussed, REC-BCD enzyme has two properties or two functions, that is nuclease and helicase both. Okay, remember it. And this REC-BCD enzyme is responsible for homologous recombination in E. coli. Okay. So, and the three different recombination systems are REC-F, REC-E and REC-BCD. REC-BCD is known as exonuclease V and it has both helicase and exonuclease activity. Okay. So we have discussed 11 sample questions so far. I guess everyone is clear with homologous recombination of in E. coli or prokaryotes, that is REC-BCD pathway, non-homologous end joining, and site-specific recombination. The recombinases like tyrosine type and serine type, and what is its function. We have discussed it clearly. If you have any doubt, then please ask me. We will discuss it again. If you want to repeat any part or any question, then also you can tell me. Or if you have any doubt regarding these topics, then please ask. Okay, so uh, let me assume then that everyone is clear with these topics and we have discussed it thoroughly. So these are some references. This is one YouTube link of homologous recombination in case of prokaryotes, that is REC-BCD pathway, compared with homologous recombination in case of eukaryotes. I think it's represented clearly and in a effective manner to understand. If you want to uh, understand it in a animated through a animated video, then you can go through this link. This is the link of homologous recombination compared to uh, eukaryotic and prokaryotic things. Okay, and this is the YouTube link of the previous session, which we have where we have discussed several DNA damage mechanisms and its repair mechanisms. If you have not attended the class, then you can go through the YouTube link to watch it over, and you can do your assignments clearly or thoroughly. Okay, thank you everyone for joining. I guess you can do your assignments properly so that we can discuss your doubts. Okay, if you have any doubts from your assignments which you have assigned, you can ask me here. Okay, any doubt from the assignments, okay, I repeat it, you can ask me here, we will discuss. Okay, so that's it for today. Let's meet again next Monday. Okay. Thank you everyone. Welcome you all. It's my pleasure. So do the assignments and ask me doubts. We will discuss. Okay. Okay.